Welcome to this fascinating lecture about kimchi and the science of fermentation. Today we're going to explore a scientific paradox. Why does kimchi stay crispy even though fermentation should soften vegetables? Here's the puzzle. Fermentation is essentially controlled rot. When bacteria break down plant material, we'd expect cell walls to weaken and vegetables to turn mushy. Yet kimchi remains remarkably crisp, sometimes crunchier than fresh cabbage. When microorganisms ferment food, they produce enzymes that break down complex molecules. This should make vegetables soft and mushy. But kimchi defies this expectation. Even after weeks of fermentation, it maintains a satisfying crunch. How is this possible? The answer involves three interconnected mechanisms working together. Let me explain each one in detail. The first mechanism involves lactic acid and pectin. Pectin is the glue that holds plant cells together in the middle lamella, the layer between cell walls. Here we see two plant cells. The orange layer between them is the pectin-rich middle lamella. This is what keeps cells stuck together. Think of pectin like the mortar between bricks in a wall. If the mortar crumbles, the wall falls apart. If pectin breaks down, vegetables turn mushy. During fermentation, lactic acid bacteria consume sugars and produce lactic acid as a metabolic waste product. This drops the pH of the kimchi dramatically. The chemical equation is glucose converts to 2 molecules of lactic acid. 1 glucose molecule yields 2 lactic acid molecules. The pH drops from about 6.5 down to around 4.0. This is a 100-fold increase in hydrogen ion concentration. Here's where it gets interesting. At low pH, calcium ions form crosslinks between pectin chains. This is called the calcium pectate gel. Imagine pectin chains as flexible strings. Calcium ions act like staples, binding multiple chains together into a rigid network. This strengthens the structure. It's like adding rebar to concrete. The calcium crosslinks reinforce the pectin network, making it stronger and more rigid than before fermentation. The second mechanism is osmotic dehydration. Kimchi is heavily salted, creating a concentrated brine solution. Typical kimchi uses 3 to 5% salt by weight. This creates a hypertonic solution outside the cells. The cell interior has lower salt concentration, shown in green. The brine outside, shown in brown, has much higher salt concentration. Water moves from low salt to high salt. Osmosis drives water out of the cells into the brine. Water always moves from areas of low solute concentration to high solute concentration. The osmotic pressure is given by the equation pi equals ImRT, where I is the Van't Hoff factor, m is molarity, r is the gas constant, and T is temperature. A 5% salt solution is approximately 0.85 molar. Let's calculate the osmotic pressure. For sodium chloride, I equals 2 because it dissociates. At room temperature, T is 298 Kelvin. R is 0.082 liters atmospheres per mole Kelvin. This gives an osmotic pressure of about 41.5 atmospheres. That's enormous pressure, driving water out of the cells. To put this in perspective, a car tire is only 2 to 3 atmospheres. This osmotic pressure is like having 14 car tires pressing on the cell membrane. As water leaves the cytoplasm, the cells shrink. The cell walls don't shrink as much, so they become thicker and more concentrated. This makes them mechanically stronger. Think of dried pasta versus fresh pasta. Dried pasta has had water removed and is much harder and crunchier. The same principle applies to kimchi vegetables. The third mechanism is controlled enzymatic activity. Fermentation is not random rot. It's carefully controlled. Normally, pectinase enzymes break down pectin. These enzymes are present in vegetables and many bacteria. They cause softening. But here's the key. Most pectinase enzymes work best at neutral pH around 7. The low pH of kimchi, around 4, dramatically slows these enzymes. This graph shows pectinase activity versus pH. Notice it peaks at neutral pH around 7. At pH 4, where kimchi sits, the activity is less than 10% of maximum. Furthermore, the dominant bacteria in kimchi, Lactobacillus species, produce very little pectinase compared to spoilage bacteria. This is why controlled fermentation is different from rot. Now let me ask an important question. If we want crispy vegetables, why doesn't boiling work? Boiling kills bacteria, so why does it make vegetables mushy? The answer is timing. When you heat vegetables, you activate pectinase enzymes before you denature them. Between 60 and 80 degrees Celsius, these enzymes work very rapidly. Pectinase works optimally around 65 degrees Celsius. By the time water boils at 100 degrees, significant damage has already occurred. Additionally, boiling doesn't create the calcium pectin crosslinks that fermentation produces, and boiling doesn't create the same osmotic dehydration effect. Let's do some quantitative analysis. How much force does it take to bite through kimchi compared to other vegetables? Biting through fresh raw cabbage requires about 15 to 20 newtons of force. That's about 3 to 4 pounds of force. Boiled cabbage requires only 3 to 5 newtons. It's become much softer, losing 70 to 80% of its mechanical strength. But properly fermented kimchi requires 18 to 25 newtons. It's actually slightly tougher than fresh cabbage in many cases. This bar chart shows the comparison. Fresh cabbage in green, boiled in orange, and kimchi in red. Notice kimchi is actually the crunchiest. Fermentation time is critical. Too short and you don't get enough acid. Too long and other bacteria take over. Optimal kimchi fermentation takes 1 to 3 weeks at 4 degrees Celsius. This slow, cold fermentation maximizes crunchiness. The pH drops gradually. Day 0 starts at 6.5. By day 7, it's 4.5. By day 14, it reaches 4.0 and stabilizes. By the end of fermentation, lactic acid comprises 0.5 to 0.8% by weight. This is about 50 to 80 millimolar concentration. Here's another interesting question. Could we get crispy vegetables by just adding acid and salt without fermentation? The answer is partially yes. Pickling in vinegar creates similar calcium crosslinks and osmotic effects. Pickles are indeed crunchy. However, fermentation produces hundreds of flavor compounds that vinegar alone cannot replicate. These include esters, aldehydes, and amino acid derivatives. Fermentation also produces beneficial vitamins like B12 and K2 that simple pickling does not generate. The bacteria synthesize these during metabolism. Let's go deeper into the molecular chemistry of what makes something crunchy. What happens when you bite into kimchi? Crunchiness is the sensation of rapid fracture. When you bite, cell walls must break cleanly with an audible crack. This requires walls to be rigid but brittle. Plant cell walls are about 30% cellulose, 30% hemicellulose, and 30% pectin. Each component plays a role. Cellulose microfibrils provide tensile strength, like steel cables in reinforced concrete. They resist pulling forces. Pectin acts as the compression-resistant matrix, like the concrete its. It resists squashing forces. Breaking a calcium pectate bond requires about 200 kilojoules per mole. This is a relatively strong ionic bond. The bond energy is approximately 200 kilojoules per mole. For comparison, a carbon-carbon single bond is about 350 kilojoules per mole. Water acts as a plasticizer. When cell walls contain lots of water, they become flexible and rubbery. Removing water makes them rigid and brittle. Can we build a mathematical model that predicts kimchi texture? Let's try a simplified approach. Texture depends on 3 main factors: the number of calcium crosslinks, the water content, and the amount of enzyme damage to the pectin. We can write texture T as proportional to calcium concentration divided by water content W multiplied by 1 minus enzyme damage E. K is a proportionality constant. For fresh cabbage, W is high at 90%. Calcium is minimal. E is 0, so T is moderate. For boiled cabbage, W drops to 80%, but E jumps to 0.7. The enzyme damage dominates, making T very low. For kimchi, W drops to 75%. Calcium concentration increases 5-fold. E stays at only 0.1. All factors combine to maximize T. Plugging in typical numbers, this model predicts kimchi should be 2 to 3 times crunchier than fresh cabbage, which matches experimental observations. What are the practical implications of this science? How can we apply these principles? First, we can improve texture in other fermented foods by controlling pH, calcium levels, and salt concentration using the same principles. Second, we can prevent mushiness in canned vegetables. Adding calcium chloride and controlling pH during canning preserves texture. Third, we can create novel products. Imagine crispy fermented carrots, radishes, or even fruits using kimchi chemistry. Some pickle companies already add calcium lactate to their brine. This provides both calcium for cross-linking and doesn't affect flavor. Let's take a brief evolutionary perspective. Why did humans develop fermentation techniques that preserve crunchiness? Throughout evolution, crunchiness signaled freshness and safety. Mushy food was often spoiled or dangerous. We evolved to prefer crunchy foods. Fermentation allowed our ancestors to preserve food without losing the sensory properties we evolved to prefer. This was a huge survival advantage. Modern food science now reveals the molecular basis for what ancient cultures discovered empirically. Korean fermentation techniques perfected over centuries have solid chemical foundations. Let me discuss common mistakes that ruin kimchi texture, now that we understand the chemistry. Mistake 1 is using too little salt. This fails to create sufficient osmotic pressure. The vegetables don't dehydrate properly and stay mushy. Mistake 2 is fermenting too warm. At 20 degrees Celsius instead of 4 degrees, fermentation races ahead, but pectinase damage also increases dramatically. Mistake 3 is not adding calcium-rich ingredients. Traditional recipes include radish and fish sauce, both high in calcium. Skipping these reduces cross-linking. Remember, optimal salt is 3 to 5% by weight. Too much, and it's inedible. Too little, and texture suffers. For those interested in deeper chemistry, let me explain pectin structure in more detail. Pectin is a polymer made of galacturonic acid units linked together. Each unit has a carboxyl group that can bind calcium. In fresh vegetables, 60 to 70% of carboxyl groups are methylated. These cannot bind calcium. During fermentation, pectin methylesterase enzymes remove some methyl groups. This demethylation exposes more carboxyl groups for calcium binding. Paradoxically, this controlled enzymatic activity increases the capacity for calcium cross-linking, even though other enzymes are breaking down pectin elsewhere. Let's compare kimchi with other fermented vegetables around the world. Sauerkraut uses similar lactic acid fermentation, but typically has less calcium and lower salt concentration. It's moderately crunchy, but not as firm as kimchi. Indian pickles use very high salt and oil. The oil creates a hydrophobic barrier that changes the texture mechanism. They're crunchy, but in a different way. Japanese tsukimono encompasses many types. Some use pressure fermentation, which physically compresses the vegetables, creating density and crunch through mechanical means. Let me summarize the key scientific principles we've covered today. Principle 1. Low pH from lactic acid enables calcium-pectin cross-linking, which dramatically increases cell wall rigidity. Principle 2. High salt concentration causes osmotic dehydration, removing water and concentrating cell wall components for mechanical strength. Principle 3. Controlled fermentation at low temperature and pH minimizes destructive pectinase activity while allowing beneficial acid production. These three mechanisms work synergistically. Each one alone helps, but together they create the exceptional crunchiness that makes kimchi unique. The paradox is resolved. Fermentation is not random decay. It's a carefully controlled process that actually builds molecular structure even as it transforms the food. This illustrates a beautiful principle in food science and chemistry generally. Sometimes the path to preservation and quality involves processes that seem destructive but are actually constructive when properly controlled. Fermentation exemplifies how chemistry, biology, and culture intersect. Ancient food preservation techniques contain deep scientific wisdom that we're only now fully understanding at the molecular level.